Hello everyone, this video is going to be going over glycolysis and gluconeogenesis. Now gluconeogenesis just ends up being sort of the reverse of the glycolysis chain, but with these four enzyme key differences. But first we're just going to run through glycolysis as an overview. So glycolysis is always going to be starting off with glucose, and we're going to be in the first step phosphorylating it. So we get glucose 6P, and this is done by the enzyme hexokinase. And of course, in this step, we're going to be utilizing an ATP to get our phosphate group. So that is going to be also utilizing ATP, giving off its 1-phosphate to yield ADP. Now in the second step, we're taking our glucose molecule and we're actually converting it to fructose. So we go from glucose 6P to fructose 6P. This time we're going to be using the enzyme phosphohexoisomerase. In the next step, we're going from fructose 6P and we're adding another phosphate group onto it. So we're going from fructose 6P to fructose 1,6P, or fructose 1,6-bisphosphate. And the enzyme that's going to be responsible for this is going to be phosphofructokinase, or as I abbreviate it, PFK. And of course, since we're adding a phosphate in this step, we're going to be utilizing another ATP. So ATP to ADP here. And moving on, this next step, we're going to be taking our biphosphated fructose and splitting it in half. And so we require an aldolase to do this. And this yields us two 3-carbon molecules. And we actually get two different 3-carbon molecules, but I'm going to simplify this because they eventually both end up being glyceraldehyde 3-phosphate. And we get two of them. So we run it through our aldase, and we take our glyceraldehyde 3-phosphate and we actually add another phosphate to it. So we end up with 1,3-bisphosphoglycerate. And the enzyme responsible for that is going to be glyceraldehyde 3-phosphate dehydrogenase. So I'm just going to abbreviate that glyceraldehyde, GA, 3-phosphate, so 3P, dehase. And for this step, we're going to be utilizing NAD, and we're actually going to be creating an NADH. Now for the next step, since we've gotten our biphosphated glycerate molecule, we're going to be popping off one of those phosphate groups in order to create an ATP. And this leaves us with just a 3-phosphoglycerate. And the enzyme responsible for this is going to be phosphoglycerate kinase. And like I said, getting 1 ATP from this. And now we're left with this 3-phosphoglycerate molecule, but we can't actually utilize that last phosphate group when it's on that 3 position. So we have to reconfigure the molecule, and in this way we get a 2-phosphoglycerate instead of a 3-phosphoglycerate. So I'm just going to abbreviate here PG for phosphoglycerate. And this is done by a phosphoglycerate mutase. So I'm just going to abbreviate this as phosphoglycerate PG mutase. we take our 2-phosphoglycerate molecule and combine it with an enolase enzyme, and this gives us phosphoenol pyruvate. I'm going to abbreviate that as PEP for phosphoenol pyruvate. And then of course the last step then is going to be yielding us pyruvate and creating an ATP molecule from that last phosphate group. So we get pyruvate. This is done with pyruvate kinase. And of course, like I said, it gives us our one other ATP. And of course, since we're doing this with one glycerate molecule at a time, and we have two of them, for every single glucose molecule we end up getting 4 ATP total, and that helps to balance out the 2 ATP we utilized initially in the earlier steps. So that's the entire glycolysis chain. What about gluconeogenesis? Well, gluconeogenesis is essentially glycolysis run in reverse. So we start with pyruvate and we build it up into glucose. But since there's a few steps that are rate limited, we need a way to get around them because they are very high energy to take off these ATP. The first major hurdle to get over is going from pyruvate to phosphoenol pyruvate. And the intermediate step we take in between is going to be to turn pyruvate into oxaloacetate. But it's a little bit trickier than this because oxaloacetate is going to be made within the mitochondria of a cell. So this first blue asterisk here is indicating that we're going into the mitochondria to create oxaloacetate. But glycolysis and gluconeogenesis mostly takes place outside in the cytosol. So what we have to do then is take this oxaloacetate that's inside the mitochondria and convert that into malate. And malate can be taken across mitochondrial membrane, indicated by this asterisk, and it can be taken back into the cytosol and turned back into oxaloacetate. So now we go from pyruvate into the mitochondrial matrix to create oxaloacetate. Oxaloacetate gets converted to malate. Malate gets taken across the mitochondrial membrane again, outside into the cytosol, and then there in the cytosol it can be converted to oxaloacetate, and oxaloacetate in the cytosol can then be converted into phosphoenol pyruvate. So there's two enzymes that are involved for the conversion of pyruvate to oxaloacetate to phosphoenol pyruvate. The first enzyme is going to be pyruvate carboxylase. And the second is going to be phosphoenol pyruvate carboxykinase. So I'm just going to abbreviate that one, PEP again, for phosphoenol pyruvate carboxykinase. Now the rest of the glycolysis chain is going to be identical to the gluconeogenesis chain until we get to this step up here where we're going from a bisphosphate to a single phosphate on the fructose. And the enzyme we're going to be utilizing there is going to be fructose 1,6-bisphosphatase instead of phosphofructokinase. So this is going to be, this number 3 enzyme here is going to be fructose 1,6-bisphosphatase. And finally, our very last step, hexokinase can't go in the reverse direction because it utilizes ATP, same as the phosphofructokinase down here. So instead, we're going to be utilizing glucose 6-phosphatase. And that's the entire gluconeogenesis chain. It's just a simple matter of working around a few enzymes that normally utilize ATP for them to go forward, or they're trying to get around the mitochondrial membrane. Otherwise, they're the exact same in their reactions and their products.